Good evening everyone and welcome to my YouTube channel. So here today we are going to be looking at interphase, the cell cycle in general, and mitosis as well as meiosis. So what is the cell cycle? So the cell cycle is a series of stages that involve cell growth, DNA copying, and also the formation of two identical daughter cells. So the cell cycle is mostly associated with mitosis and interphase. So just as we said, it involves the cell growth, the copying of DNA, and also the division to produce two identical daughter cells. So let's look at the first stage of the cell cycle. So under the first stage of the cell cycle, we have interphase. So interphase occupies most of the cell cycle. About 90% of the cell cycle is mainly interphase. So now interphase is part of the cell cycle which involves the cell growth and also the replication of DNA. So just as we said, it involves cell growth. It carries out normal activities and also replication of DNA. So that is mainly what interphase is all about. So this is done in preparation for division. When we look at division, we are looking at we can talk about mitosis. Now let's get into details. So let's look at the interphase involves G1, S phase. Now before it goes to S phase, it might go through what we are going to call G0, which is this stage, before it goes to the S phase, which I'll explain. We also have the G2. Then from G2, it will now enter the M phase, which is mitosis or meiosis, like that. So that is interphase. So now let's get into details of interphase. So what are some of the things that occur during G1? So G1, just G1 is we refer to it as gap 1. So under G1, you can just think of G as growth. So the series of events that take place here are: number 1, growth. Not only that, we also have the creation of important organelles such as the mitochondria, which is used for energy. So there is the creation of organelles and also just enzymes that may be used in DNA replication. We also have, this is all happening in G1. We also have the creation of ribosomal RNA. So these are made during G1. So growth, creation of important organelles such as mitochondria, and also the creation of enzymes, or rather the synthesis of enzymes that might be used during DNA replication, like that. So this is what we are going to call G1. So growth and all these are happening. Now the cell does not necessarily just move from G1 direct to the S phase. So it depends on certain conditions. So the cell first might be able to either go direct to the S phase or return to G0. Now, why return to G0? So now, G0 is referred to as a resting or quiescent phase. So quiescent simply means the same as resting. This is where the cell is more like in its resting stage. So it's just a fancy way of saying resting. So now you might wonder to say, why does the cell go to G0? So the cell moves from G1 to G0 if these conditions are not met. Number 1, when the cell has got, we would say that lacks enough nutrients. We can also talk about if it's lacking growth factors. Growth factors are the factors that cause or stimulate the growth of the cell. Lacking growth factors, there are other things such as the need for repair. Like for example, we might be looking at the DNA. The DNA is already there in G1. Now, if the DNA is damaged, there is need for it to be repaired before there could be synthesis, because we don't want DNA that is damaged, something like that, to be copied again. That would be more like it would cause a genetic malfunction and all that. So if there is need for repair, a G0 might be entered, need for repair. For others, so these are some of the reasons that might cause the cell to move from G1 to G0. There are also other cells that just enter G0 permanently, where they are not dividing. The examples of these that we might have are the neurons. So these are fully more like, well, or rather the biological term, they are fully specialized. So there is no need for them to return from G0. So these enter G0 permanently to maintain genetic stability. So these enter G0 permanently, the neurons. Other cells that just enter G0 temporarily, we might be talking about the liver. So the liver enters G0 temporarily. So yeah, that is what you need to know about G0. So it is a resting stage. So now during this period, the cell might be preparing itself for cell division, in the sense that if it lacks nutrients, it increases its nutrients. And also if the DNA is able to be repaired, it repairs the DNA so that it can re-enter the S phase. Then we also have, when it's lacking growth hormone, there is need for the growth hormones to be stimulated. And if it's lacking nutrients are more like supplied to the cell so that it may now be able to enter the S phase. Now if you look at this is for those that undergo temporarily G0. These will be able to, if there's the repair, there will be repair growth factors introduced and also some nutrients added, then it will now be able to enter the S phase. But before that, before it enters the S phase, we need to know the examinable checkpoints. So when we look at this, we have at this point where it is, we are going to realize that there should be a checkpoint. So now how does the cell detect that the nutrients are sufficient? That is where the checkpoint comes in. So we are going to call this one as our G1 checkpoint. So the G1 checkpoint is the checkpoint that occurs when the cell wants to enter the S phase. So as you can see, this is the restriction point, meaning to say this is where the cell detects whether it can enter the S phase or not, or it should go back to our G0, which we talked about. Now, this checkpoint detects whether the DNA is damaged or not. If the DNA is damaged, it simply means to say it cannot enter into the S phase, because in the S phase there is the duplication of DNA, but we don't want damaged DNA to be able to be copied. So we don't want something like that to be able to happen, the DNA that is damaged to be copied, which would cause a mutation. So now this checkpoint is very, very important. It also checks the size of the cell. So if the cell size is not sufficient, the cycle will not move to the S phase like that. Also, just as we talked about the growth signals, if they are not there, the cell will still not be able to enter the S phase. So this is very, very important, especially for the DNA, if it is alright. Now, when all these are met in terms of the DNA is not damaged, the size of the cell is sufficient, and growth signals are present, the cell will be able to enter what we are going to call the S phase. So the S phase, you might just think of S as synthesis. So during the S phase, what occurs is DNA replication. Also, not only DNA replication, but also the centrioles are duplicated. So if you recall, centrioles are the ones that help in formation of the spindle fibers, which help move or rather separate the chromatids, which we'll later discuss. So now during S phase, this is what is happening. So we have, let's say we have our chromosome here. So the chromosome is copied, duplicates itself, or rather is copied. So as you can see, it forms more like an identical part of it. So this is what is happening like that. So the DNA begins to copy itself like that, or rather the chromosome begins to copy itself like that until it makes the other part that is identical to itself. So this is why the checkpoints are important, because we don't want the DNA that is not well to copy itself. So not only that, but also centrioles, which form spindle fibers, are duplicated. So as you can see here, this is our mother centriole like that. It forms the daughter centriole like that. So there is duplication. So these are the ones that occur in our S phase. So as you can see, those are our centrioles, which look like that. So it is important for them to be duplicated so that they may move on opposite sides. Let's move on. So now when we look at our G2, under this one, we are going to have further growth. Other organelles are also checked, or rather duplicated organelles that might be used in the later stages. We have a number of things that occur here, and also there is now what we are going to be calling the G2 checkpoint. So the G2 checkpoint, make sure that the DNA is correctly duplicated. So when we look at the DNA, the way it is, it's like this. Now the G2 checkpoint makes sure that the DNA is correctly duplicated. The other part is exactly the same as the original part. Then that's the G2 stage. So it is very, very important for this to take place, because it makes sure that even as there is copying of DNA and all that, there is the division of these chromosomes, the two created have got an equal distribution of DNA, or rather the chromosomes. So now this is how interphase occurs. Now we are going to move to our next stage. Now if the cell has not correctly copied the DNA, then we are going to find that the cell cycle will be halted, meaning to say it will be caused to stop. Now this is controlled by the TP53 gene, which we are going to look at later in this video. From there, let's look at what we are going to call the mitosis. Then from mitosis, we go to meiosis. So now let's go to mitosis. What is mitosis? So mitosis is simply the process by which a eukaryotic cell divides into nucleus to produce genetically identical daughter cells. So now let's break it down. So now, just as I have said, so this process occurs in eukaryotic cells. So the eukaryotic cells, we might be looking at humans, among other things. So now in this one, we are going to have the division of the nucleus. Then there will be the formation of two genetically identical daughter cells. That's mitosis, each having the same number of chromosomes as the parent. That's the way it is. So if, let's say, we have the parent there, then it will have the daughter cell there. So in other words, when they say that each with the same number of chromosomes as the parents, they don't mean to say there will be two daughters that will be produced. No. Even the parent itself, at the end of it all, becomes what we are going to call the two daughter cells. So there's nothing like at all, this is the parent, from the parent, then there will be two. No. Just one is formed, then both of them, including the parent, will be called the daughter cells. So that is what we mean by mitosis. So now the mnemonic that we are going to be using for mitosis is what we are going to call PMAT. So according to PMAT, we are going to have Prophase, we are going to have Metaphase, we are going to have Anaphase, we are going to have Telophase. Then when you look at your this mnemonic, metaphase, you can think, since it's starting with M, you can think of middle. So at this point, chromosomes align at the center. Then anaphase, you can think of it as away. So chromosomes are pulled apart. So you can think of the word away like. So let's look at our prophase. So prophase involves a series of stages. So the number one thing is that the nuclear envelope breaks down. So this is more like the membrane of the nucleus. So this breaks down. It breaks down like that. Then we are going to also look at our nucleolus, which is this dense part of the nucleus. This one disappears. Then the other thing is that we are going to have, when these disappear, we are going to have the chromosomes condense. So chromosomes condense. So what is the exam question that you might be asked? So they condense so that they might have this structure that you can see. So this is this condensed form of a chromosome. So they might ask you, why do chromosomes condense? So why do chromosomes want to look like this? So the reason is very, very simple. Number one reason why chromosomes condense is firstly to prevent tangling. To prevent tangling. So this is very, very important in the sense that when you look at a chromosome, the chromosome in a cell might be as long as 2 meters, and 2 meters is very, very long. So now chromosomes condense. So let's say we have this one chromosome, we have another chromosome like that. So now if chromosomes are just like this, then they can easily be able to tangle themselves as they move like that, and of which it is not required for chromosomes to be entangled with another chromosome, as this would cause other issues. So chromosomes have to condense to avoid them from being tangled. So tangled where they more like intertwine, of which intertwine where they wrap around each other. Now this is not required, as this would promote breaking. So if they tangle themselves, they might end up breaking, because this chromosome wants to go this other side and this chromosome wants to go this side. So as a result, they might end up being more like breaking apart. So to prevent that, to prevent the breaking apart, what we have is that chromosomes condense so that they can, number one, prevent tangling themselves and also prevent breakages. The other thing is that when they condense, they easily move. So we can say easy movement. So as you can see, when the chromosomes are in their condensed state, they can easily move from one point to another. So we could have something like that. So they can easily be moved from one point to another if they are in their condensed form. Also, when chromosomes have condensed, there could also be easy separation. Easy separation, or rather easy or accurate, let's use the word accurate separation, because after cell division each part has to more like be separated. One part goes the other side and the other one goes the other side. So this is made possible because of the chromosomes condensation, meaning to say they can accurately be separated equally so that each cell has got the same number as the other part. So to continue our prophase, we are going to have these structures that we are going to be calling our centrioles. Centrioles move to opposite poles and then they produce what we are going to be calling the spindle fibers. So the chromosomes are X-shaped. Now when the chromosomes are looking like that, they are now called sister chromatids. So sister chromatids are simply the chromosomes that are found that are attached at the centromere like that. So these are what we are going to be calling the sister chromatids like that. So now this is how prophase is. So now if you just know what prophase is, it will be very easy for you to look at telophase, because telophase is just the opposite of it is just the opposite of prophase. Let's move on to our metaphase. So metaphase, from the word midu, these chromosomes will align themselves at the center like that. So the center is what we are going to call the metaphase plate. It is also known as the equatorial plate. So when they align themselves at the center, then we are going to have the spindle fibers, which were released by the centromeres. They will attach at the center of the chromosome like that. So at all points, the spindle fibers will attach at the center like that, at the center like that, at the center like that. So this is what happens under metaphase. Now, under metaphase, we are going to now look at what we are going to call the M checkpoint, which is the metaphase checkpoint, also known as the spindle checkpoint, also known as the, so generally just the M checkpoint. So the M checkpoint makes sure all chromosomes are attached at the centromere by the spindle fibers and are at the metaphase plate. So it is the M checkpoint that ensures that each chromosome is attached to the spindle fibers and aligned at the metaphase plate. So that checkpoint is very, very important. You should remember it for even your exam questions. Now, why is this important? M checkpoint is very, very important as it prevents aneuploidy. Aneuploidy. So now this is very, very important. So now aneuploidy occurs where there is an even distribution of chromosomes. So when the chromosomes are aligned at the center, what we require is that each chromosome has got half the number. Now if the spindle fibers are not correctly attached, this may happen. So we might have a situation whereby they are supposed to move to opposite poles, but one of them is taken to one side. Now this would create this could be one of the causes of cancer because there is an even distribution of chromosomes. So now it is very, very important for the spindle checkpoints to take place so that there is the prevention of aneuploidy, which is the uneven distribution of chromosomes. So now remember, the metaphase, it only involves the spindle fibers attaching to the chromosomes that are aligned at the center like that. So this, where we have the separation, that's anaphase, which we are going to look at now. So during anaphase, just remember what we said. Anaphase, you can think of away. So under this one, we have the separation of the daughter chromosomes. So these sister chromatids, as we call them, so these are chromatids. So these are chromatids together. So this and this forms what we are going to call chromatids. So these are pulled apart during anaphase. So this is done with the help of the spindle fibers that have been attached. So when there is a separation which we have shown there, then that is our anaphase. Let's look at our telophase. So when we look at telophase, remember, it is just the opposite of prophase. So under telophase, the nuclear envelope reappears. So in prophase, it disappeared, but here it shall reappear. Then these ones, the spindle fibers break down. Then we are also going to have, then the centrioles return to their original position. So these are our centrioles. So remember what we have said, the spindle fibers break down, the nuclear envelope reappears, and also the nucleolus also reappears. The nucleolus reappears. Then we are also going to have the cytoplasm starts to divide. Then we are going to have our brief stage, and also let's look at our chromosomes. Our chromosomes begin to decondense, meaning to say they unwind. So they begin to decondense. So this is how we refer to as telophase. So telophase is basically the opposite of prophase. So if you know prophase, just say exactly the opposite for telophase. So now let's look at cytokinesis. During cytokinesis, we are going to have the separation of the cytoplasm. So as you can see, when the cytoplasm separates, where we have the cytoplasm in here and the cytoplasm in here, it's more like new daughter cells are formed with the centrioles there. We have chromosomes that have condensed, meaning to say they unwind and all that. So this is cytokinesis, which is just the splitting of the cytoplasm. Let's move on to just the importances of mitosis. So the importances of mitosis are simply for: number one, asexual reproduction. So some will undergo asexual reproduction under this one, where there will be the formation of a daughter cell just from the parent. It is also important in repair, as new cells are replaced during mitosis, because mitosis leads to the creation of identical cells. So let's say if this is the skin, the creation of another skin will, let's say here if there's a bone, the creation of another skin will allow for it to be repaired. We'll also have another thing, growth, because growth is the permanent increase in the dry mass as well as the number of cells. So mitosis leads to the creation of new cells. So in other words, mitosis occurs in all somatic cells. Somatic cells, we are simply referring to them as body cells. So that is some of the importances of mitosis. Another one is regeneration, which is more like just healing. Regeneration is also done during mitosis. It is also important for the maintenance of genetic stability. Now let's move on to our meiosis. So meiosis is just the type of cell division that reduces the chromosome number by half, producing 4 genetically unique daughter cells. When we look at this, we are going to have cell division that reduces the chromosome number by half, producing 4 genetically unique daughter cells. So they are unique, meaning to say they are different. That's why, for the sake of the exam, meiosis is called reductional division. Then we are going to call mitosis as equational division. So mitosis is reductional. Since there is the reduction of the number of chromosomes by half, then for meiosis we are going to call it equational. So it is called equational because there is no change in terms of the number of chromosomes. So the number of chromosomes are identical to the parent, meaning to say it is sustained. So equational division has to do with mitosis, then reductional has to do with meiosis. So let's get into it direct. So after interphase, then we are going to have meiosis. So let's get into it. Meiosis has got two of them. We can say 2 PMAT, in which case it will have Prophase I. So we said this is Prophase, this is Metaphase, this is Anaphase, this is Telophase. Now let's begin with our Prophase I. So Prophase I is slightly different and more complex as compared to mitosis prophase. So firstly, we are just going to have the same things that we looked at. The centrioles move to opposite poles, which we talked about. So centrioles move to opposite poles. They produce spindle fibers. The nuclear membrane disintegrates, or rather disappears. The nucleolus also disappears. So you can, if you are taking notes, you can just write the same things there. Now the difference comes in. First, the other same thing is that the chromosomes are condensed. So they condense there. Now the difference comes where they pair up. So the homologous sister chromatids pair. So when we are looking at homologous chromosomes, these are chromosomes that carry the same gene but are coming from the mother, one coming from the mother and the other one is coming from the father, like that. So these pair up like that. So when these pair up, then we are going to call this one a unique name, which we are going to call synapsis. So synapsis, in other words, is just pairing up. So synapsis is pairing up. So when these pair up from the mother and the father, then we are going to call it synapsis. So this synapsis, which is just the pairing up, when they pair up now, they will be able to join together to form what we are going to call crossing over. Now you may see this word might come. We have tetrads, which is just a pair of homologous chromosomes. So in other words, when you have this one is a sister chromatid to this one. So when we have 4 of these, because this is number 1, number 2, number 3, number 4. So this is what we are going to call a tetrad together. Tetrad meaning 4. So when these are 4 of them, then we are going to call them tetrad. Or in other words, just a pair of homologous chromosomes. So homologous chromosomes are carrying the same gene, one coming from the father, one coming from the mother. So when these come together, this process is called synapsis, which is just pairing up. Then they will be able to do crossing over. So this crossing over involves where the mother's chromosome and the father's chromosomes attach together at the points that we are going to call the chiasmata, like that. So we are going to have something like this. So at this point, this is what we are going to be calling a chiasmata, like that. So yeah, let's just reverse it a bit. Okay, so this point is what we are going to be calling our chiasmata. So the chiasmata is the point of exchange. So now what begins to happen is that as these two genes separate, after that it's more like when they separate, what begins to happen is that they exchange genetic material at that point. just you might just think of it as when they are trying to more like let go of each other, they tends to damage each other in such a way that the part that belongs to one chromosome remains on the other, and the part that belongs on this chromosome remained on this one. So this is what we basically refer to as crossing over. They attach there, then they will be able to separate. When they separate, one will carry from the will carry genetic information from the mother and the other one from the father, like that. So this leads to variation. So this we call this term where we have the portion that is carrying genes from the mother on, let's say if this is the father, then this part is for the mother, and this one, let's say this one is for the father, then we have the mother, like that. So this one we are going to just call it a fancy word which we are going to call genetic recombination. So genetic recombination is simply the fancy way of saying that the chromosome carries genes from both the mother and the father due to crossing over. So recall, due to the fact that chromosomes are carrying from the mother and the father, this leads to variations, meaning to say the differences in the traits between the offsprings and the parents. From there, let's move on to our next stage, which we are going to call Metaphase I. So Metaphase I is the process by which the homologous pair, which the chromosomes, one from the father, one from the mother, align themselves at the center side by side. So they will pair along the metaphase plates like that, side by side. So now remember, under metaphase 1, you're going to find that yes, they are aligning side by side. The thing is that although they are aligning side by side, each one carries a portion from the mother and a portion from the father. So if I can zoom in this, you will find that there is some green part here, which you will realize here, and there is some blue part there. So this is genetic recombination. So when they align themselves at the metaphase plate, the spindle fibers will be able to attach at their centromeres. So the middle part of the chromosome is called the centromere, like that. So now when they attach there, then there will be the spindle checkpoint, which is just checking whether the chromosomes are correctly attached to their centromeres. So all these, even here, it will be able to be attached at that point. So all these chromosomes will be able to be attached like that. So this is happening and all that. So this is very, very important. So even the difference is that the chromosomes align themselves side by side, while in meiosis or in mitosis, it will just be on one line, so just one like that. But under meiosis, they will align themselves side by side like that. So let's move on to our next stage, which we are going to call anaphase. Anaphase, think of the word away. So now under Anaphase I, we are going to be having the separation of the homologous chromosomes. Now take note, as you can see 4 things. We have one here, 2, 3, 4. So in other words, it's just that they have changed the shape, but they are just separated like that. So it will be moved from one side from the middle to the other side. So we are going to have something like this. Let me create an imagery. So this is how they are moved. So they are moved to opposite sides but are not separated. So that is what happens in Anaphase I. So after Anaphase I, we are going to go to Telophase, which is just the opposite. Under Telophase I of meiosis, so you are going to find that the nuclear membrane reappears Also the nucleolus reappears like that. Then we are going to have the centrioles move to their original sites. Then the spindle fibers, which were those things, they break down. Then the chromosomes remain like that. Now we are going to have the cytoplasm starts to divide at that point. So it starts to divide, and this is completed during cytokinesis. Then we have cytokinesis 1 of meiosis, where the cytoplasm is split into two. We have the nucleolus has reappeared. We have everything, two diploid daughter cells. So they have these chromosomes. The only difference is that this one has got both the maternal, or rather from the mother, and from the father, which is our paternal, like that. So after cytokinesis 1, one thing that I forgot to mention that is very, very important when we are looking at metaphase, we are going to have chromosomes will align themselves at the they will align themselves side by side like that. Now this is done in a random order, which we are going to be calling as random assortment. Sometimes it is called independent assortment. So this is our random assortment. So this also leads to many variations, because we don't know which side in terms of assortment. We don't know which side the chromosome will be. Like for example, if this is the mother chromosome, we don't know if the mother chromosomes will be here or here and here and here. Then we have the father there. So it does not necessarily dictate which chromosomes are going to be on which side. That is what we call random assortment. So the chromosomes align themselves in a random order. This brings about variations because you will have many traits, like just from the mother on one side, just from the father on the other side, or we could have them mixed like that. So that is what we call random assortments. This also increases the variations. So basically that is what random assortment is, where they extremely align themselves at the metaphase plates in a random order. So sometimes when they ask you, they might talk about independent assortment. Don't confuse it with Mendel's law of independent assortment. When they ask you, compare which one has got the highest influence between independent assortment and crossing over, you will take independent assortments to just be random assortment, because chromosomes will align themselves independently of the other. We might have mother's chromosome on one side, both of them, the father, the mother, and all that. So due to this, there are a lot of many possible arrangements. Remember, there are about 46 chromosomes in total, which will more like align themselves. We have 23 on one side, 23 on the other side. There will be a lot of different combinations. Now due to this, if you are asked to compare which one has got a greater influence between random assortment and crossing over, the correct answer is random assortment, because of the many possible combinations that can be made possible. We might have from the mother, the father, and all that. There are a lot of many possible combinations that can be made just from chromosomes aligning themselves in a random order. So if you're asked such a question, maybe it's carrying 5 or 6 marks, which one has got the greater influence between crossing over and random assortment, you first begin by defining what crossing over is, what random assortment is, then add a comment. However, the one that has got the greatest influence is, you are going to say, is random assortment, because there are many possible combinations that can be made in terms of arrangement at the metaphase plate in comparison to the crossing over, which generally affects the genes. Now, without wasting time, let's look at Meiosis II. So Meiosis II is very similar to Mitosis. So I'm going to split the video just in case you haven't watched Mitosis, you can refer back to that. So now Meiosis II is just the same as Mitosis. Let's just run through. So Prophase II of Meiosis is just the same as the prophase of Mitosis. Nuclear envelope breaks down. We have the centrioles move to opposite poles, they release spindle fibers and all that. Then what we are going to have is that so that's basically it. So just as we have said about mitosis, that is what happens in prophase 2. So now same with metaphase 2. Let's look at it. Under metaphase 2 of meiosis, so this one just involves the chromosomes will align themselves at the center. So the same with mitosis, metaphase, they will align themselves at the center. The centrioles will attach at the centromeres and all that. Now, one common exam that they might ask you is, what is the difference between the metaphase in mitosis and metaphase in metaphase 2. So before this, let's look at the similarities. So the similarities is that they both align at the center in one line, just as you can see. Now there are two cells due to the first cytokinesis one. So we have cell 2, we have cell 1. So all of this is happening. So they both align at the center in both metaphase of mitosis and metaphase 2. They both align at the center. The difference is that if you look at the chromosomes that are at the center, these are recombined, meaning to say they are carrying genes from both the mother and the father. This is why you can see a different color at that point. So that is the difference between metaphase in mitosis and metaphase 2 in meiosis. This is due to the fact that crossing over occurred. So anaphase 2, the separation of the daughter chromosomes. So we have something that is this. So here under anaphase 2, remember under anaphase 1 it wasn't separated, but anaphase 2 it is just separated, which is the same as mitosis. So we can look at it and realize to say these reactions are occurring at the same time. So two cells are undergoing like that. So when we refer to telophase 2 of meiosis, so telophase 2 is where the opposite of prophase, there is the reappearance of the nuclear envelope. We have the centrioles move to their original point. Then we have the decondensation of chromosomes. Then we also look at the cytoplasm. It starts to divide like that. Then finally, we are just going to have our cytokinesis 2. Cytokinesis 2 of meiosis involves just the formation of 4 haploid cells. So as you can see, the haploid daughter cells just has got more like 23 chromosomes instead of the usual 46 that humans have. Each cell will have 23, 23, 23 and 23. So this is what we refer to as the half number of so the daughter cell has got the half number of chromosomes. So that is what happens. It forms 4 cells like that. So that is what meiosis did. So this is how the sperms and the ovum mostly have, or rather have, 23 chromosomes, so that when they fuse together, they are going to add up the 23 from the sperms plus the 23 from the ovum. Then they'll be able to create a zygote which has got 46 chromosomes, thereby maintaining the number of chromosomes. So these variations, only one of these cells can be able to more like combine with a female cell like that to form a zygote. So that is meiosis. One last thing, let's talk about the gene that I told you, the TP53 gene. What does it do? So the TP53 gene is a gene that codes for the p53 protein. So what is its major function? So the major function is that it plays a role in regulating the cell cycle, DNA repair, and the process that we call apoptosis. So just as I have said, the role in the cell cycle, the role is the p53 protein helps in regulating the cell cycle, DNA repair, and apoptosis. So apoptosis is just programmed cell death. So you must know this for your exams. Now let's more like clarify more on the p53 protein. So our p53, which is coded, the TP53 gene is more like attached to the chromosome. So it's a gene already, so it's attached to the chromosome. So it is able, during duplication, or rather replication, the gene is supposed to look like this part, but it has, let's say, replicated like this. So what will happen is that the TP53 gene will be able to detect that the DNA has been incorrectly copied, and therefore it will stimulate the production of the p53 protein, which will be able to more like do one or two things, which will call, firstly, it will try, let's say for example, if the chromosome is like this, then the replicated chromosome is like this, where we have something like this, then we are having something that here it's missing. So what it will do is that it will stimulate the process of repair, where this part that is missing will be repaired like that. So it will be repaired, then the cell cycle would continue. But if, let's say, for this situation whereby the DNA is or rather cannot be repaired, if it's looking like this, let's say it has got a lot of things and it cannot even be repaired, then what will be stimulated, is the process of apoptosis. So what is apoptosis? Apoptosis is simply what we are going to call programmed cell death, where the whole cell itself dies, so as to prevent it from affecting the whole organism. So it just dies. So that is what we call programmed cell death. So this gene is very, very important. It regulates that the DNA is not damaged and all that. That's why it is called the guardian of the genome. So yeah. Now, the TP53 gene is coming from the TSG, which means the tumor suppressor gene. So it is the tumor suppressor gene. So it is a tumor suppressor gene. So being a suppressor gene, it controls or reduces uncontrollable division. So it controls, in other words, controls uncontrollable, or rather prevents is the best term, prevents uncontrollable and prevents uncontrollable division. So the one that controls the uncontrollable division is what we are going to call the tumor suppressor gene, of which P53 is an example, or rather the TP53 gene is an example that prevents uncontrollable division. So now this leads to the prevention of tumors. So this leads to the prevention of tumors. So a tumor is more like the accumulation of mass like that. So when there is a lot of uncontrollable divisions, there will be the creation of a tumor, because there will be a lot of mass that will be created. So it helps in the prevention of that by controlling or acting as brakes for the cell cycle. So it helps in acting as the brakes for the cell cycle. Otherwise, this works under the checkpoint. So whenever there is a checkpoint, it makes sure that it halts, or rather stops the cell cycle at that point, so that the things can be done to prevent the whole organism from suffering. Thank you very much for watching. Hope you would like, subscribe, and share for more. See you in the next tutorial.